Thank you very much. So I'll be talking about fighting for DNA justice, a genotyping software in the Nick Hillary acquittal. Let me first tell you what genotyping is. Genotyping is the concept of being able to make a comparison between two items. If you look at the top of the slide, if I want to compare item A with item B, I can't make a direct comparison if I'm looking at a piece of clothing and a person. There's no way to make a DNA comparison or genetic comparison. So a laboratory takes that item and translates the item into data, which you've been seeing for the last 2 days, short tandem repeat data or other DNA data. And most methods of forensics directly try to compare data with data. And that leads to a lot of problems and a lot of the issues we've seen over the last 15 years. You can't do that in any rigorous mathematical way. It's just basically wrong. So what ends up happening is the data need to then be converted into a genotype. That is, what is the genetic type that's determined from the DNA profiles. And then it's legitimate scientifically to make comparisons between the genotype that has been derived from one item and another item. They can be evidence items that you're comparing, or they can be an evidence item with a reference item. What happens when there's more than one genotype that appears? This is where the word probabilistic genotyping comes in. All it means is that when there are multiple possibilities, you have to do something in terms of assigning weights or probabilities to that more than one possibility. And that's how the uncertainty in the genotype is represented, is through probability. That's what you have in most of life and most of science. Usually you don't get one exact definite answer unless you're in theology. Usually in science, there's multiple possibilities, and in all of science and law, there are different probabilities that are attached. And then a comparison can be made between the probabilities of those, say, 10 different outcomes and, say, one reference sample or more possibilities. So each of these represents a genotype, and you can make comparisons between the probabilities of the genotype, and the math works exactly the same way. How do you get those probabilities that I'm showing here in the middle as a posterior probability? The way it's done is one begins with a prior type that's shown on the left. That's what's in the population. That's the type of a random person. And then there's data, the STR data, and using math, the prior probability is updated with that middle column of the data, and it's called a likelihood. And that updating of the data, then you just multiply it together and then make the total come out to 1. That's what gives these probabilities that we then have comparisons with. I think that's it for the math here. But that's Bayes' theorem. You start with some initial belief, you see data, you update it through a likelihood function, you have new belief or a new type, and then you can make a comparison in order to get some sort of a statistic. So over the years, there's been some pretty bad methods of interpreting DNA mixtures. The concept initially with thresholds was that if thresholds worked very, very well with single source DNA, why wouldn't they work very well with mixtures? And the reason is that you don't have one or two peaks of the same height. It's much more complex. You can have taller peaks, shorter peaks, and across the different possibilities of different sizes that are indicated by the boxes on top, there are different intensities, which are the relative fluorescence units, indicating more DNA for an 8 and 11 peak, or less DNA for a 12 and 14. So instead of dealing with large amounts and small amounts, tall peaks, short peaks, patterns, what was done 15 or 20 years ago was just carrying over this concept of threshold, taking a scissor to the data, cutting through it, and saying anything that survived that scissor cut is in, anything that was too short is out. It's kind of like if you take your bank balance, if you make more than $300 a year, it's the same as if you're Bill Gates. That's what thresholds do. The idea was to throw out data, and now you'd have a list of whatever these values were, the alleles 8, 11, 12, and 14. What CPI did is it took it one step further and said that for genotypes, there would be no weight of different probabilities. Every possibility that was on a pair would have the same weight. So it would be all or none. Either an allele pair genotype was present or it was absent. And the result was a tremendous loss of information, a lot of error. You've heard how studies from the federal government starting in 2005 began showing these methods give wildly different answers in different hands. And in fact, there have been a lot of papers on them. I added to this literature a year ago with a paper. The title is Inclusion Probability, that's CPI, for DNA mixtures is a subjective, one-sided match statistic that's unrelated to identification information. Basically, it's a random number. You count up the number of tests. For every 2 tests, you get about another factor of 10. That's all it ever was. And so millions of these DNA mixtures have been done for the last 15 years that have had no probative value. All they do is they confirm with some number the subjective impression of a DNA analyst. So the hope is the next 15 years won't look like the last 15 years. There's another phenomenon called dropout. Dropout is true in nature. We have dropout. You can have events that are alleles dropping out. So if you have lower amounts of DNA, you may not see them in a signal. They may not appear in electropherogram. They may be under a threshold or just not there at all. The phenomenon is there, and there are mathematical ways of addressing it by considering all possibilities. But a very interesting shortcut is to just introduce a parameter that you can tune. See the stereo knob? You just turn it up when you want to handle more drop. So what is dropout saying? The data that you wish you were there, in order to convict somebody, isn't. So just turn up the knob, and then mathematically it's back. Since you might not believe that, I'm going to back that up with some data, some experiments we did on some dropout programs. And here, we're turning up the knob from left to right. We're just increasing the dropout parameter and saying maybe more alleles have dropped out. The vertical axis is the match statistic expressed in log units. 0 is 1, 6 is a million, and -6 is 1 in a million. And as you turn the knob up, the match statistic goes from exclusionary, -6, which is 1 in a million, gradually up to 6, which is a million. So just like you can change thresholds to change your data and get whatever answer you might want, you can also play with dropout parameters. A dropout parameter is not the phenomenon in nature. It's a tool to dial in an answer. FST here in New York is one such method that used dropout. And as you know, the legal aid DNA unit was instrumental in precluding that in a recent case. So I'd now like to move on to New York versus Nick Hillary. This is a rough map. I guess, given that we're in New York, the center of the world is in the bottom right. But up in the hinterlands, on that red dot, all the way near Canada, is Potsdam. And then we'll also enter into the story in the bottom left is Pittsburgh, Pennsylvania, where Cybergenetics is. So on October 24th of 2011, Garrett Phillips, age 12, died from strangulation. And an immediate suspect was Nick Hillary, who was a local soccer coach at the college, and he was arrested for the murder of Garrett Phillips a year and a half later. DNA evidence was collected, actually quite a bit of DNA evidence, ultimately about 150 items. But this is a listing of the evidence items, about 35 of them, that were initially processed by the New York State Police. There was clothing, the shorts that the deceased was wearing, a long sleeve shirt that he was wearing, and a black long sleeve shirt that was taken from the accused apartments looking for blood from the victim. There were also tiles on the floor that had different biological items in them. There was a theory that whoever was fleeing from the homicide jumped out of a window, so DNA was taken from the window and the screen, the sill, and the area around that point of possible exit. There was also DNA taken from the victim's body. There was an oral swab taken, DNA from each of his hands, from his thumb, and from his neck. So that's the starting point of biological evidence that was collected. We were sent this data, so I'll tell you a bit about Cybergenetics. Cybergenetics is in Pittsburgh, Pennsylvania, beautiful city, no steel, no smoke, just a lot of eds and meds. That's what we do. Cybergenetics was founded in 1994 after I'd been at Carnegie Mellon in computer sciences faculty for 10 years. And what we do is we extract information from genetic data. We've worked on cancer research, medical diagnosis. In fact, we reanalyzed data from the World Trade Center, all 18,000 victim remains, comparing them with missing people. Most of our focus for the last 5 or 10 years has been on DNA mixture analysis, how to extract information from DNA mixtures. So in June of 2013, the New York State Police Lab, with whom we were working in other projects, sent Cybergenetics the Hillary DNA evidence for us to screen. We always begin by looking at evidence at no cost from anyone who sends it to us. It's usually prosecutors, defenders, or police. I guess it could be someone else, but that's usually who has DNA evidence, to see what information might be there. And the focus from the lab was, they said, please look at the left-hand fingernail scrapings. So that's what we began looking at. And we looked at it in TrueAllele. TrueAllele is a technology that over 20 years ago solved the PCR stutter problem for genetic markers, also in forensics. We solved the mixture problem in 1999 and published that in the Journal of Forensic Sciences 2 years later. And then we began developing a much more statistical approach, building on that mathematics, coming up with a probability for every conceivable genotype without looking at what an answer might be. And that went through 25 versions of refinement for about 10 years on data sets that we kept testing on more and more complex mixtures, until finally it had really completely exceeded what its creators could ever do and the people we work with. And we felt it was reliable and began use in criminal cases in 2009. I won't give you a long lecture on TrueAllele. There are other talks you can find in papers. I'll just summarize its main features. It's accurate. There have been 34 validation studies, 7 of them published in peer-reviewed journals. It's objective in that its workflow, as we'll see in a second, removes human bias. It's been accepted the last, 6 years. There have been 10 Dormer or Frye challenges, and TrueAllele's done fine. It's transparent. We provide to either side, both sides, we really don't care. The answers are the same. A 4 gigabyte DVD that explains how TrueAllele works with papers and mathematics and videos. We give the software to whoever wants to run it in a criminal proceeding. Either side can rerun whatever they want for free. It's just there as a resource. And it's also neutral in that it can statistically include or exclude. Why is that important? Because the usual methods, like combined probability of inclusion for mixtures, like CPI, can only include. So the notion that the computer doesn't care, you're going to get a number. If it's a bigger number, like 1 million or 1 billion, that's support for inclusion. If it's a smaller number, like 1 in a million or 1 in a billion, that's statistical support for an exclusion. And that's reflected in the workflow. The workflow is we begin with the data. The data comes in the form of files that come from the end-stage machines of a crime lab, and they're sent to us by defenders or prosecutors or police. And those files are put into a computer. It's all entered. There's no choice. Everything's just entered and used. It's uploaded to servers that then separate out genotypes, calculate them. Afterwards, comparisons are made, and the math decides. There's not a person making a decision here. After those separated genotypes are compared with references or with other evidence, either there's a big number suggesting that someone's in the DNA, or a very small number suggesting that someone is out of the DNA. And this accords with the National Academy of Sciences report from 2009 of keeping people out of the process with robust software that eliminates human bias. What did TrueAllele find in Hillary? Well, initially we only looked at the fingernails. And In 2013, the lab hadn't done the usual 1 or 2 tests for some reason. They were very interested in the fingernails, and they had unusually done 26 identifiable tests on fingernails. What we found was a mixture that was about 95% of the victim, 5% of someone else, with no statistical connection to Hillary. The numbers were slightly exclusionary, but pretty much inconclusive in terms of making a definitive answer. And we advised the lab to use a kit designed for degraded DNA called Minifiler to get data for degraded DNA that would permit a scientific resolution of the question of who else might be in there. One of the reasons why, we'll see data in a second, of why there was inconclusive results is that the genotype of Nick Hillary was very similar to either the genotype of Garrett Phillips or some of the stutter artifacts around. And therefore, more possibilities had to be considered that could be inclusionary or exclusionary. A year later, the lab had some new kits and new machines. They sent us another 9 tests, and they asked us to look at it. And again, we found no connection to Hillary. We sent them an email, and that was communicated with the district attorneys who were in the process of changing at that time. So at this point, we've helped the police, we've helped the prosecutors, we're working pro bono, and there's no connection. There's a more recent type of software called STRmix. It comes out of New Zealand. As you'll see on the bottom, the company that makes it, ESR in New Zealand, it's a commercial company. You can go on their website, and this is from their 2016 annual report, where they're pleased with the financial results of increasing profits and expanding into international markets like New York City. STRmix does keep the old concept of threshold. It keeps the old concept of dropout, and in this way it introduces and continues to maintain subjectivity in the analysis. It does add peak heights, so instead of getting rid of the data completely with thresholds, instead the thresholds determining at what level you begin to consider the peaks with their heights. It throws out all the evidence underneath the threshold. It also does some statistical modeling. It doesn't do a vast amount of modeling. Modern statistical systems in all fields, like TrueAllele, spend most of their time measuring their own uncertainty. Simpler systems don't. It also requires calibration on data because it doesn't have all of that modeling and all those variables, so it needs to fill in what those values are from other data. It solves easy mixtures quite well. If you test it, you'll see on simple 2 or 3 person mixtures that don't go much below 20%, you get nice answers. In the workflow of a lab, it's a good replacement for CPI, the PopStat software from the FBI. It gives much more accurate numbers within that limited range, and it retains the conventional workflow. So this is the workflow that crime labs have used for 15 years on DNA, particularly when there's uncertainty, as with mixtures. The first step is you pick your data. You pick which peaks you want to use. There are guidelines, you have thresholds, but there are thresholds, there's dropout, there's ways of picking which tests, which locations, and which peaks you want to use. We'll see some of that. After you choose your data, then a human expert decides if your guy is in there or not. That's the process. It's written in the protocols. If you do discovery and you read them or contact legal aid for a copy, you'll see a person decides, and then after it's been decided that the person is guilty, or not guilty, I guess, included in the DNA mixture, then you can get a statistic to confirm that expert decision. And it's interesting because you might call it step 1, then you do step 3, you determine the answer, and then you do step 2, which is to use a computer. That's the workflow that we see for PopStats from the FBI the last 15 years, as well as for STRmix in the current protocols that we've seen from laboratories. And then, of course, you don't tell anybody that that's what you're doing. It's kind of important. The result is you put people back into the process, and that overcomes the limitations of the software of not knowing what to do with the low-level peaks, which is why there's thresholds and dropout parameters in the first place, and that it can introduce human bias. So in January of this year, these were the New Zealand results that were obtained. The 3 developers of the STRmix software were consulting for the prosecution. They used version 2.3. They found a match statistic of 10 million connecting Phillips' fingernails to Hillary's data. Under other assumptions, when they removed forward stutter peaks, making some subjective decisions about the data, they could reduce that statistic to 10,000, and they decided that the amount of that unknown contributor that was connected to Hillary was less than 0.5%, so less than 1 part in 200. It's probably worth noting that no system has really been validated extensively down there. Having worked with the police and the prosecutors, Cybergenetics continued its pro bono efforts, now being contacted by the defenders. And the Legal Aid Society of New York contacted Cybergenetics. They sent over the STRmix declaration, which was the report, along with the initial discovery. That had a lot of stuff in it, but none of it was terribly helpful. We worked with them to do a more extensive discovery in which we got more useful information, including the computer results file that described exactly what the computer had done. And that was quite important. And that was the key to seeing how they chose data to get the results that they got. Here are some of the ways that data are chosen. First, let's talk about thresholds. This is a study that was done 2 years ago by NIST together with the Netherlands Forensic Institute. It was presented at a conference in the Netherlands. And what it showed is that if this is the match statistic, starting off at 1 trillion, as the amount of DNA is decreased for a 10% minor contributor, the match statistic falls off. The blue line is TrueAllele, leveling out to maybe 1 billion or 1 million or so. However, programs that use thresholds, and here the cutoff was at 50 RFU, and Dropout, and these programs were STRmix, LRmix, and LabRetriever, rapidly lost information and were down at 0, to which the developers attributed this to thresholds. In fact, they contacted people afterwards and said, you can't use a threshold of 50. You should be using this with a threshold of their software with a threshold of 30 or 25, and that would be a fairer comparison. This is probably the most important slide. Independent experiments were done on an evaluation copy of this version of STRmix, and what was done was using the choices made by the prosecution expert, ESR, under one scenario, as well as removing all the forward stutter. The threshold was varied. Think of it starting at 80, 70, 60, 50, which is what was reported, 40, and 30. And you would think that statistical software, has the word statistics in it, would be very robust and not sensitive to small variation. So here's the reported number. At a threshold of 50, we have a number in the tens of millions that purport to link the defendant to a crime. If you raise the threshold to 60, it drops to 250. If you raise it to 70, the number drops to 30, and finally at 80, it disappears completely. If you lower the threshold, because why is there a threshold in statistical software anyway, just 10 RFU. The scale goes from 0 to 10,000. So a change of 10 RFU is 1 part in 1,000. He's not there. He's excluded. And if you drop it to 30 RFU, he's not there. So at 40 and at 30, he's not there. And even under these other stutter assumptions that were made with lower numbers, he's rapidly not there. As thresholds are raised, lower numbers, then at 40, he's gone, and at 30, he's gone. This is what we were looking at and wondering what this was doing in a criminal court. These are some examples of how data are chosen. These are different alleles. This is data. The arrows mean that those peaks go up about 10 times taller. The different blue colors are 3 different experiments that were analyzed together in STRmix. And you see there's a lot of data here. There's a lot of blue bars. Here's a threshold of 50, 100, and so on. Those are the different RFU levels that could be set. 50 was used. This is a visual representation of what was in one of the STRmix reports. VV is the victim. So at 10 and 12, there's the victim, the very tall peaks. He's 95, 99% of the sample. There's a foreign allele, F, that was indicated on the table. and that's at allele 16. And here's the defendant, which was listed in the third column, having a genotype DD of 12 and 16. And based on that, clearly he's in there, if that's how you choose your data. But if you look at all the data and you start looking either at peaks that people could say, oh, that's just stutter, let's not use that. Oh, maybe that's stutter, let's not, oh, we don't use that. That's after stutter, oh, we're not forward stutter, we're not going to use that. That's too low. It's kind of like Casey at the bat, right? You just keep watching the data fly by. Every one of these question marks is a potential allele, and any pair of those should be considered. But in a nice little table, it looks very incriminating because you're not looking at the original data and the choices that are being made. There's another fascinating thing that we saw about dropout. I'll summarize it by saying there are about 15 alleles at a given locus. There's about 100 allele pairs. The dropout method is very popular with software developers because it means you only have to look at 3 or 4 alleles, and you're not looking at all 100 possible pairs. You're only looking at maybe 10 pairs. Everything's a lot faster. But the problem here, and I'll summarize this for you, is, for example, at locus D18, there were, in this table from the STRmix report on the 3 amplifications, they chose 4 of the alleles over threshold, 9, 10, 11, and 12. All the other alleles are represented by Q, meaning something else that we're not seeing. So Q in something is something you have seen with something you haven't seen. And QQ means it's not in the data you're even inputting it into the program at all. It's just not there. And they gave that a 15% probability. But the bottom line is this. Hillary's genotype is a 1717. It's not one of the alleles that they're looking at. So his genotype is not in the data that they put into their program. And so what they're comparing is the 1717 of Hillary with the QQ of the data that isn't there in order to get a match statistic bigger than 1. I think you understand what I said, so I'll move on. How are you getting inclusionary statistics from somebody who's excluded from the data? Legal Aid asked us to do a software comparison. I think we looked at 60 different attributes for CPI, FST, STRmix, TrueAllele, looking at categories of data, model, match, human involvement, the process, the system. This is one of them. For human involvement, for these different categories, software is basically driven by people choosing their data in subjective ways, and that's the key distinction between TrueAllele, which doesn't have people playing with the data. Then in March, because we were really curious, we took all the samples we had. Nobody asked us to do this, but we just, hey, we're working pro bono anyway. We have the data. We're scientists. We don't know what's going to happen. We did evidence-to-evidence comparisons of all 35 items to all 35 items, determining to what extent are there positive associations or non-associations. Most everything was exclusionary without an association, and also compared to references. This is what I had the foresight to take a photo of on my iPhone because I've long since lost the sheet of paper. And so I graphed the associations. And what we saw was that Nick Hillary was associated with a cluster of his own shirt taken from the apartment. We didn't know what it was at the time. We checked with Legal Aid to say, is this incriminating evidence? No, it's his own shirt. Of all the different clusters, so Nick is not associated with the fingernails or any of the other parts of the victim's body, but there is an association between the different parts of the body and with the victim's own shirt. So his neck and his hands, his thumb, they're all associated. That's this cluster here. And it's either the case that this is an artifact of the victim's own profile, or it's actually another person who's there. We did some sensitivity studies. We think it's more likely another person, but there's no proof of that. It could just be an artifact. Whatever it is, it's not associated with the defendant. And it appears on all these evidence items which the police and their consultants and the prosecutors were not looking at. They're only looking at the fingernails instead of trying to get a sense of the whole picture. In April, there's another report from New Zealand. ESR has a new version, 2.4. They've added after stutter modeling, which as best as we can see is if you put in the peaks and you have extra peaks there with fewer contributors, the program doesn't crash. It may do something else. We really don't know. But now the match statistic is at the level of 100,000 with this new software. However, looking at the STRmix report, the 10-page listing of what the program actually did, we still see they're using a threshold of 50 RVU, and they turned up the dial on dropout. They just turned it up a little bit. You still included if you choose the data in that way. There were mixture validations that were discussed as legal aid was getting ready for requesting for and ultimately having its Frye hearing for admissibility. This one came from the District of Columbia. It was done in December of last year. And what the graph is showing is that as the peak heights change from, say, 0 up to 2,000, the average peak height, this is the match statistic. Above the dashed line is inclusionary, with numbers going from 10 billion and higher, exclusionary 1 in 10 billion and lower. There's two interesting things. The first is on these two-person mixtures, the program is either including some, now the other thing to say is the circles on top are when they know somebody's present, and the orange circles on the bottom, so there's blue circles on top, the person's truly in there. On the orange circles on the bottom, the person's not in there. And when the person's not in there, you almost immediately just get negative 30 log units. So this program is not very good for subtly working out the degree of exclusion for exonerating people. It just says you could view that as exclusion or fail. For inclusion, it's very gradual. Here's just a rapid fall off. But this region, which Clint Hughes cross-examined one of the analysts on at some length, is worth a close-up, which is what was presented at the hearing. So what we're doing is we're taking the small region, and in the area from 0 to 250 as the peak height, we're zooming in, and on this scale, this is around 1 million, that's 1 in a million, and this is 0. Here's the point. In the range of the data in this case, where we're looking at peak heights between 50 and 100, here are match statistics in the range of hundreds and thousands. Those are the values. And you can't distinguish the orange from the blue. Is the person really there, blue, or are they really not there, orange? You get the same number. So this program, by this validation study, isn't terribly helpful in resolving those questions. You just get some low inclusionary number, which is a false positive for the orange cases and a low number for the blue cases. To confirm this further, in June, there was another report. The New Zealanders just kept sending reports, and they did their own validation study back in Australia, New Zealand. Here, true is a 0, and somebody who's really in there. False, somebody who's not in there is an X, and they're not really distinguishable. I'll zoom in again, but here's the same kind of scale. Here now what we're measuring is the quantity of DNA. One cell is 6 picograms. So here's 5 picograms. So that's 1 cell, 2 cells, 3 cells, very small amounts of DNA. This is the level of half a cell, which is about 1 part in 200. So what's happening right in this region? Well, zooming in again, this is a range. The X is larger match statistics of people who aren't in there, and the X's. The O's are people who are there. And this is the range we're talking about, 1 in 200, 1 in 250. And in that range, going up to, say, 5 is 100,000 is a match statistic, we can't separate out the X's and the O's, whether the person is really in there, an O, or the person's not in there, an X. So we're not getting any discriminating power for separating out who is getting a positive. And think of it like a medical test. For all the men out there, right? You get a positive PSA. What does it mean? Everybody gets a positive PSA, right? Do you have the disease? Don't you have the disease? There are a lot of tests in medicine where they have a high false positive rate. And so when you get the test, a positive number isn't determinative of whether you have the condition or not. In this case, it's not determinative of is the person present or absent from the sample at this low level. There was a Frye hearing. Legal aid was successful in getting that in July. Fascinating transcript, which perhaps Clint Hughes would be happy to share with you. And they kept coming back to this question of 30 RFU, where validation studies have been done for STRmix, and where if you, between that 30 and 50 RFU range, there's a lot of peaks that would produce an exculpatory statistic. So the question is, why aren't you looking down there? And so Mr. Hughes asks, did you have any curiosity about what the value would be if you ran your program at 30 RFU instead of at 50, where you're throwing out all this evidence? And the response from the ESR expert was there was really no point. They'd have to do a lot of work and go back to the beginning to extract the peaks from the gene mapper program. So with the data they had, they'd have to do more. And he said, and yes, indeed, I am curious. In fact, I'd like to go down to 10 RFU. As much as there was curiosity, there was actually no software that was run, that was reported on, to satisfy that curiosity. Well, companies can offer independent defense testing, maybe this is from a form provided by ESR, the corporation in New Zealand, to provide access to STRmix software by a legal defense team. And that was requested by Hillary's defense, and there was agreement on the terms and conditions, and as far as I know, the software was never obtained. It was never received, so in principle, if we hadn't had this evaluation software that we could test the program on, nobody would ever have known about any of these decisions that were being made. In August this summer, STRmix was precluded, and this is just taken from Judge Felix Catena's decision. And there was a concession by the prosecution expert that there was no internal validation study done by the crime lab that generated the data. Why is that important? Because in the words of the judge, the expert was forced to pick and choose the data from different sources and pick and choose the input parameters. How much threshold? How much dropout? So all of this was subject to subjective decision-making, where a small difference in parameter or data had a huge impact in inclusion or exclusion in the end. And so the court ordered that the defendant's motion to preclude be granted because clearly with such a high sensitivity to parameters and data without an internal validation or calibration or any of the things that are needed for the software, it would be hard to say that it was completely reliable. There are some interesting out-of-court statements that the New Zealanders continued to make while the trial progressed after STRmix was thrown out. They did ultimately say that they ran STRmix on the fingernail data at 30 RFU, but in order to do it, they had to assume more contributors and do more with drop-in order to get an inclusionary match statistic. That is, the question of running the way you ran it in December and January with 30 RFU and more data, instead of at 50 RFU and less data, what did you get? Well, we know what they would have gotten, but they're not really talking about it. They acknowledge that STRmix has a need for these thresholds since a small difference in the machine that's used has a large difference in the outcome. That's not an issue in TrueAllele. We've run it multiple ways, but we model the baseline noise, so we don't need thresholds. They did acknowledge that they were pleased that the judge found STRmix to be reliable, which is a statement in that the method is reliable, though they said that the judge did preclude it for other case-specific reasons that were not delineated. It was also, as worth mentioning from a cultural perspective, that they suggested that the main reason that Cybergenetics had asked for better data, this mini-filer data, that's designed for degraded DNA. When DNA is chopped up, there are kits that can get at the shorter fragments that are chopped up. Mini-filer is one of them. Their thought was that the reason you ask for more data is to support the prosecution's hypothesis. We must have believed Hillary was in there and guilty. This goes back to the philosophy that we saw in the workflow. The TrueAllele workflow, which is pretty standard in science, is you get your data, you put it into a computer, it objectively does whatever it's supposed to do, and when it's done, you get a number. And that number tells you to what extent somebody is present or absent. Big numbers mean someone's really in there. Very small numbers, 1 in a trillion, mean they're really not in there. And in between, you have less information. But the usual workflow in forensic science for the last 15 years, and perhaps for the next 15 years, is that's not what you do. You pick your data. After you choose it, you decide on the answer. And after you decide on the answer, you take the data that you chose along with the person who you've made the decision on, and then you run a confirmatory program. So it's clear from that worldview that the only reason we'd ask for more data would be to confirm the answer we already had. But that's not our workflow. That's not how we approach the science. We want the data to be determining what the answer is. So last month, Nick Hillary was acquitted on September 28th, and that was an interesting outcome in that it took a huge amount of work to get this acquittal for one person for DNA with this newer software. You have to imagine there's a team of 5 people back in Pittsburgh working at Cybergenetics. Our computers and servers are running full tilt, adding up all the evidence we looked at in the end, about 150 items and the work that was done. We're a small company of 10 people. This is about a $250,000 pro bono project. So we can't do this for everyone. And I believe that Legal Aid of New York City had a similarly sized team also working pro bono on this case. So this is the amount of effort you have to do to undo what crime labs are moving toward. That's a heck of a lot of work. Hopefully there's a better way. What's good about this case is that it gives us, the New Zealanders at ESR, the STRmix people, gave us a very good roadmap of what you need to do to proceed and succeed. First, you need a full discovery. You can't mess around. What they give you, what we see, and everybody sees right now, is not enough for you to do much with. You need the original data, you need their reports, and this is described in more detail in one of the materials on your USB drive. This is science. The only way you're going to defeat questionable science is with science, and science means testing with either this method or some other method. You have to test STRmix on case data. You're going to have to get hold of it. Otherwise, you're not going to be able to do a sensitivity analysis and see when it fails. Sometimes it gives a good answer. Sometimes it doesn't. You have to compare with accurate results. You need to use some other program that may be more robust in order to determine what the answers really are. This may be new for some defenders. But Brady is on your side. You really want to know what's true if you're going to fight what might not be true. You need to identify where the subjectivity and bias crept into these reports and challenge unreliable results, less in terms of Frye hearings than in terms of the discovery and the cross-examination of the deficiencies. You don't want to go into every county on easy cases that any good software would get an answer on and challenge reliability. You want to identify the bad cases, like in the Hillary case. And there's a treasure trove of documents from the Hillary case with an amazing number of statements, reports, testimony, and so on. I'd like just to say a few words about PCAST, the President's report. It relates 15 years of CPI failure that was never really questioned. That might be an opportunity for civil litigators or enterprising defenders who want to go back into the past and look at these hundreds of thousands of cases. But there's another 15 more years of nonsense or non-science, and this is a time to start questioning, what do you need to do? Now, you don't need to become scientists. What you need to learn to do is how to challenge effectively, and that was a nice part of the collaboration that Cybergenetics had with Legal Aid for half a year. What's needed is legal education, and I'm in the process of forming a nonprofit to try to be able to provide the scientific legal education to all attorneys, but my guess is it's going to be defenders who will need and benefit from it the most. And you want to find truth in every case to compare it against what you have. And here again, part of what this nonprofit will be doing is performing free TrueAllele screening. We currently do it for cases that come in, but if we were deluged with 1 million items of evidence next week, we don't have the resources to do it. So this sort of free screening can identify where the information is, and we're in discussion with different cities about providing this screening is an automatic approach. You get your discovery precedent set in your city, like, for example, OCME, and then you get a constant flow of electronic data, and nobody has to look at it. It just gets piped into a computer, and you know what the results are by science that you can then compare with what the laboratory purports to be claiming as science. On your drives, there's some reading materials. I broke them into 4 categories for you. There's an index in the materials. There's some readings on the top left. You want to read the basic book chapter about what is this type of genotyping, how does it work. There's a nice perspective by a former public defender in New York, Shirley Duffy, and the Wakefield admissibility ruling for upstate New York is a nice ruling about TrueAllele. There's a list of validations that are interesting. Please don't read the math or get too heavily into it, but if you look at the pictures, read the introduction and the conclusion, you'll get a sense of what these studies are about. If you want more information, you can contact me. I think an important paper, which is supposed to be published in the spring, is a white paper that I wrote. It's about CPI mixture failure, and it's about how the CPI mixture statistic failed. It's a 30-year history. It's written for lawyers. It's gentle English. There's no math. It's history. This is what happened, and this is why you might have wanted to detect it sooner. And you want to learn from history to avoid the errors of the past going forward. I also put in a paper, which is the one that why CPI is a random number. That might be of interest. And then there's some documents about the Hillary case. And then for future reference, there's also a listing of STRmix discovery items that you definitely want to include in all your standard discovery requests, because it's hard to see how you can analyze cases without data, software, knowing subjective decisions, and so on. If you visit our website and you take off a year, this will keep you entertained with lots of talks, articles, presentations, webinars, and so on. Thank you very much.